All right, we're going to start chapter 6, and chapter 6 is where we start talking about cells, and all living things are made out of cells. Some of them are only made out of one cell. Many things are made out of billions of cells, but a cell is the unit of all living things. Okay, so they're the fundamental units, they call them, and all organisms, if they're alive or they once were alive, they're made out of the cells. The cell is the simplest collection of matter that can be alive. Kind of like if you're talking about an element, or the smallest things of matter are atoms. Well, the smallest things of living, the smallest unit of a living thing is a cell. And they're related. They came about, they can actually find or follow their history, cells. Um, they can differ depending on their job, but they share some common features. So when if you compare a nerve cell to a muscle cell, obviously they're going to have different characteristics, but there's some things about cells that are very similar. Okay, the thing with a cell is you have to use a microscope to see them because they're just too small, but you can see them with a microscope. Like when we talked about in the chemistry part, when we talked about atoms, you can't see them with a microscope, but you can see cells. Okay, so microscopes, and you will do one of these, one of those Labster things with microscopes. They are used to visualize cells. There's a couple different kinds. The light microscope is the one probably that you think of when you think of a microscope. You look through an eyepiece, and it has the light is bent, and it allows you to see a specimen. It magnifies it. Okay, like the light microscopes, they magnify 40, 100, and 1,000, I think. And then if you use oil immersion, you can even do better. They lenses in the microscope, they have a series of lenses. They bend the light so that the image is magnified. And again, so you can see things that you normally couldn't see. Okay, there are three important parameters of microscopy, things that we use microscopes for. Obviously, one is to magnify, to make the image look much larger than it actually is. And you can calculate it. The way you figure that out is you look at your microscope and the numbers, you multiply them together, the number on your objective and the number on your eyepiece. Another thing that's important is resolution. Okay, that's the clarity of the image. Okay, you can magnify it, but if it's all blurry, it's not going to really do you a whole lot. So microscopes are really good at helping make the image look clearer. And then contrast. Okay, now you can change the contrast by usually how much light there's like a diameter underneath the microscope. You can twist it around, because if you're looking at something that's already pretty light, you're going to want to tone it down a little so you can see the differences in the things that are on your slide. So these are the three important parts of microscopy. Magnification, obviously, we want to make things look bigger. Resolution, if we make them look bigger, we still have to make sure we can tell what they are, so they have to be clear. And then contrast, we have to see the brightness, the difference in the different things that are the different organisms that are on your slide. Okay, light microscopes, yes, like the ones we have at the college, they can magnify 1,000 times, so they can make something look 1,000 times larger than it actually is. Okay, there are other techniques that you can use to make it enhance it even more. Okay, you can stain the components. If you've ever done the lab where you take onion cells work really well because it's easy to peel off a little piece. And you usually stain it with an iodine stain, and that will make the cell walls darker so that you can see different parts. Or sometimes you can see the organelles better if you use some dyes like besides iodine, there's methyl blue, there's some other ones that help you see the parts so that it's not just, one big blob. Okay, the resolution of a standard microscope, you can't really see organelles. When you look at a cell with a regular microscope, you can see the membranes, you can see the nucleus, and that's usually about it. Okay, they're just too small, the organelles are. So then they have better microscopes, fancier microscopes, more expensive microscopes to help so that you can see the organelles. And an organelle is a little organ inside the cell. Okay, and we're going to talk about all of them, but basically they're like we have organ systems in our body. We have our respiratory system and our digestive system and all that. So the organelles kind of serve like that, but they're inside the cell. And obviously, if they're inside a cell, they're pretty small. Okay, so When you look at a cell, one of the things you can see is the outer membrane, the outer boundary, if you want to call it that. Okay. And so when we're looking at cells, we can look at that outer boundary, and we'll talk about that actually in a whole chapter. But when we look at cells, we actually have two different kinds. We have, or two types, I should say. The two types are what they call a prokaryote and a eukaryote. Pro meaning before, eu meaning true. Okay, so a prokaryotic cell, they're much tinier. They're not fully developed, if you want to call it that. And then the true cells, the eukaryotic cells, are the fully developed ones that have all the stuff that they're supposed to have. Okay, there's only a few types of organisms of prokaryote. There's actually more prokaryotes and eukaryotes because one of them is bacteria, and you can fit a lot of bacteria in a little space. So if you're doing sheer numbers, there's more prokaryotes. But if you're doing types, there's not very many different types of prokaryotes. Bacteria and archaea, another name for that would be like blue-green algae. Okay, they consist of prokaryotic cells. All right, most of the things you think of when you think of a living organism, protista like algae, fungus, all the animals, all the plants, all of those are all eukaryotic cells. So when we talk about living things, most of the time we're talking about eukaryotic cells. Okay, so if we compare them, both of them have the plasma membrane, the outside wall. I'm checking the time, sorry. So they both have like a boundary that separates them from the rest. They have inside, they have something called crystal, or we call it cytoplasm. It's a semi-fluid substance what everything sits in and all the reactions occur in. Okay, they do have what they call chromosomes. Chromosomes are like the little X things that are made out of DNA, and they're made out of a protein. And they have genes. So they have these chromosomes, but they're not organized in a nucleus like we have. And they have ribosomes. Ribosomes are little tiny organelles that make proteins. Okay, so both types of cells have this. Okay, but the prokaryotic cells, theirs isn't really organized. Like when we look at our chromosomes, our DNA and all that stuff, it's in a nucleus. It's in the brain of the cell, the center of the cell that has, well, like I said, the brain. Well, a prokaryotic cell doesn't have a nucleus. Okay, so their stuff is just kind of floating around. It's not as organized. Okay, their DNA is unbound. Okay, it's kind of floating around. They call it a nucleoid, so it's kind of like a precursor to the nucleus, but it's not as organized. It doesn't do the same functions. They don't have organelles. They do have ribosomes, but they're not membrane bound. In other words, they're not organized. And again, you'll see when we talk about a eukaryotic cell, how our organelles have membranes and hold them together and all that. Not so much in a prokaryote. And the cytoplasm is bound by the plasma membrane. In other words, it keeps it in there, which is, something they're missing one less. They have one less layer of that fluid than we do. Okay, if you look here, here's a bacteria, and honestly, a bacteria is about the size of our organelles, so it's pretty small. Okay, there's your nucleoid. You can see it's not very, it's just kind of a line here. This area, it's not as organized as our nucleus is. This is rod shaped. Okay. They have a flagella, which is a tail that helps them move. They have a little capsule around. Okay, so they have some parts, but they're really the precursor to the true cell. Okay, eukaryotic cells, true cells, they have DNA in a nucleus. Okay, it has a membrane that keeps the DNA in the nucleus. It's really important that it stays there because remember, DNA is your blueprint to build you. We need to keep it in a safe. And the safe is the nucleus. So the nucleus has a membrane that keeps it in. Okay. All of the organelles have membranes. All right. They make sure that the stuff that's inside that organelle stays inside and then, doesn't float around in the rest of the cell. The cytoplasm is the region between the plasma membrane and the nucleus. So they have cytoplasm. They have this fluid where they call it cytosol. It's not quite the same consistency in a prokaryotic cell. And a eukaryotic cell is much larger. Like I said, if you look at the organelles in a eukaryotic cell, they're about the size of a prokaryotic cell. So you can fit several prokaryotic cells inside a true cell, a eukaryotic cell. Okay, so those are the differences. The prokaryote is like the precursor. They're just so much smaller and they don't have all the organization. And again, these are bacteria mainly. Okay. The plasma membrane is an outside barrier. Okay. It's like the cell, it's called a cell wall, a cell membrane. We won't call it a cell wall because cell walls are only in plants. But what it is a special barrier, and it allows things to come in and it allows things to go out, but only certain things. So the plasma membrane is like a security gate. Okay. You have to go up to the gate and get permission to go in or out. And the plasma membrane is the one that controls that. Okay. And we're going to actually, there's a whole chapter on this, we'll talk about it, but you can see they have the wall itself is 2 layers of what they call phospholipids. Okay. Remember, lipid is a fat. We have different ends that line up this way. Okay. They have a hydrophilic, which likes water, and a hydrophobic that doesn't like water. So if stuff that has water wants to get in, they have to count on these proteins to help. But anyway, it's set up. I'm not going to spend a lot of time talking about this, but basically what the plasma membrane does is it's a security gate. And it allows some things to come in and some things to go out. If the cell is making hormones, those hormones need to get in your bloodstream so that they can go wherever they need to do their job. So the hormone's made, and then it comes in here and it's like, hey, I gotta go do my job. Oh, here you go. Boom. These guys, here's carbohydrates because the cell needs some sugars so it can make energy. Oh, yep, come on in. So that's their job, is to control what goes in and out of the cell. Okay. One thing about cells is they can only be a certain size. All eukaryotic cells are similar in size. And the reason for that is we have to have this exchange between the inside and the outside. If we get too much stuff inside, there's not enough outside layer to take care of it. So it's kind of a surface area to volume thing, okay? So they can only grow to a certain size. And if they get bigger than that, then they can't regulate. The plasma membrane is too small to regulate what's trying to go in and out. The gate is too small. So instead of building a bigger gate, which they can't do, they'll divide and get 2 more smaller cells and start growing again. But they can only reach a certain size. If they get too big, there's too much surface area, and there's not enough surface area for the volume inside the cell, and they can't regulate what goes in and out. Okay? So as it increases in size, the volume grows. But the volume inside grows faster than the surface area. So that's why cells can only grow to a certain size, and then they have to quit because they either have to, that's it, or they divide and go smaller and then start growing again. Because again, the plasma membrane can only control so much fluid. And when the inside gets too big, then it can't control it, and then the cell's going to die. Okay? So here's an example of a eukaryotic cell. This is an animal cell. Some of the parts, and we're going to talk about all the parts individually. Here's that plasma membrane. Okay, here's the nucleus. We have something called the endoplasmic reticulum, kind of a cool word, right? And there's smooth and there's rough, and we'll talk more about that. We have the Golgi or the Golgi. It depends on who tells you that. I've heard it both ways. There's lysosomes, mitochondria. Mitochondria are really important for energy. Perioxum, they have all these things. There's a microvilli. There's all kinds of things. So we're going to talk about these individually. Okay, so I just wanted to show you this picture. And then here's a plant cell. The difference again now is a plant cell has a cell wall. It has another outer covering. And the reason for that is, you think about it, plants don't have bones. They need something to give it structure. So their cells actually have an extra layer on the outside to give them structure. If you look at plant cells, they look like bricks. They're kind of all stacked up. If you look at animal cells, they're kind of scattered because we have bones and, we have skeletons to help with our structure where plants don't have that. So that's why trees get so tall and all that good stuff. Okay. So I'm going to end here, and then we'll start talking about the organelles individually on the next one. But we got a good start on what cells are. Make sure that you do your assignment that follows.